Good day, class. So for today, we're going to discuss about the laboratory diagnosis of viral infections. According to Mr. Joshua Lederberg, he is a famous molecular virologist or molecular biologist. So according to him, sabi niya, the single biggest threat to man's continued dominance on the planet is the virus. So technically kasi, viruses, as we have discussed in our previous module, viruses are cellular non-living things that infects their host cells. No? And wherein, kapag ini-infect nila yung cells na yun para mag-replicate sila, in the process, sinisira nila yon. And eventually, ano nangyayari sa patient? They become debilitated o kaya, worst case scenario, namamatay sila or nanghihina sila. Okay? So, yun yung main reason why it is the single biggest threat to man's continued dominance on the planet. Okay? Our first topic is the clinical virology lab, significance and relevance. So here are the examples or the significance or the relevance of the clinical virology lab nowadays. So these are the reasons why we need to learn or we need to have the proper knowledge on the clinical virology laboratory. So number one, for the virus-specific antiviral drugs. So basically, viruses are very difficult to destroy or to eradicate, no? So basically, clinical virology or virology laboratory or research laboratories, ang purpose nila or objective nila is to produce virus-specific antiviral drugs. Say for example, yung recently na nangyari na pandemic caused by the COVID-19 or the coronavirus, no? If hindi nakapag-research or if hindi nag-exist ang virology laboratory or clinical virology studies, hanggang ngayon, nasa pandemic pa rin tayo. So, yun yung significance niya. So, basically, for research then, no? Number two, rapid diagnostic techniques. So, if hindi natin madadiagnose ang isang viral infection, especially yung mga rapid testings, kapag hindi pa na nagawa itong mga to, matagal bago malaman ng mga physicians nyo na may viral infection ng mga patients natin. Number three is for cell lines for cell culture. So, later on, didiscuss din natin yung topic na to, no? And then, fourth is the real-time polymerase chain reaction. So, basically, this is the one that you are studying in your molecular biology, the PCR, no? And lastly, novel viruses. Pag sinabing novel viruses, so these are the viruses that exist na bago, no? They are considered as new or novel, yun yung tawag sa kanila. Pwede rin yung mga emerging na infections. Okay. So next, these are some examples of viral diseases which requires laboratory diagnosis. Number one dyan is STIs. STIs or natawag nating, so natatakpan siya, no? STIs. So, STIs are yung mga sexually transmitted infections. Especially dito sa Pampanga, sa Manila, sa Olongapo. So, common ang STIs na viral, bacterial, tsaka mga fungal STIs. So, in virology setting, these are important for lab diagnosis. The second is diarrhea. Pangatlo is respiratory diseases caused by viral infections. Pangapat is aseptic meningitis. Okay? So, those that infect our CSF. Okay? And panglima, we have the arbovirus encephalitis. And then congenital diseases, hepatitis, and lastly, those that are viral infection in immunocompromised hosts. So this will be the main topics that we are going to discuss for today: laboratory diagnosis, Number 1, we have the cytological or histological diagnosis ng viral infections. Electron microscopy. Pangatlo, the virus isolation in cell or tissue culture. Fourth is the direct detection of viral antigens or genes. And panglima, we have the molecular diagnostic methods or the nucleic acid testing. And lastly, the serologic detection of viral antibodies. And syempre, before we start, we need to first consider yung viral specimen considerations. Kung ano yung mga kailangan nating malaman sa pag-collect tsaka kung kailan natin ko-collect yung mga viral specimens. So, ayan, no? So, specimen selection and collection. So, basically, tulad lang ng other infections sa microbiology, sa bacteria, tsaka fungi, it depends sa test, no? Depend sa test. Okay? Dun sa test performed. Say, for example, the test is regarding nucleic acid testing ng HIV. So, basically, ang sample mo would be serum. Example, ang testing natin would be reverse transcriptase polymerase chain reaction for COVID-19. So, ang sample mo would be nasopharyngeal swab or yung mga saliva, ganun, no? So, it depends sa test. Okay? Another reason is sa ating pag-select ng specimen at pag-collect na rin is depend dun sa tissue na tina-target or yung cell na tina-target. So, according sa Module 1, ano kasing tawag natin dito? Pag target tissue or target cells, ito yung tinatawag na cell tropism. Okay? So, yan yung kino-consider natin whenever we collect specimens for na viral origin. Okay? Next naman is when. When do we collect these specific specimens? So, according to the books, no, especially kapag galing kayo pupunta kayo sa ibang bansa wherein there are multiple seasons. So, depende sa season. So, we have what we call seasonal. So, there are viruses na nagpipick sa winter season, sa summer season, etc. Dito naman, locally, we have also those viruses na nagpipick sa summer, such as yung rabies. Meron ding nagpipick towards after ng rainy season, no? After ng tag-ulan, kapag nagiging stagnant na yung mga tubig, which is yung dengue and chikungunya, no? So, yung mga virus na yun, they are seasonal. Another consideration sa pagko-collect nyo is kailangan as early as possible following the onset of symptoms. So, later on, di-discuss natin yan sa serology, no? So, we collect specimen as early as possible following the onset of symptoms. Ibig sabihin, pag lumitaw pa lang yung pinakaunang sintomas niya ng kanyang sakit, no? Kumari, linignat siya or inuubo siya, that is the best date, okay, para mag-collect ka na ng specimen niya. Okay? Now, after you collect the specimen of your patient, okay, what do we do na? Okay, what's the next step? So, specimen transport and storage. So, usually, there are what we call reference laboratories wherein kailangan mo pang ipadala yung inyong specimen para magkaroon ng confirmatory result. Okay? For example, pag nagkaroon ka ng positive HIV screening sa laboratory mo dito sa Pilipinas, no? According to Medtech Law, kailangan mo siyang ipadala sa RITM, which is the reference laboratory, para ma-confirm na HIV positive siya. So, kailangan mo siyang i-transport. So, anong dapat mong i-consider doon? Number 1, it should be refrigerated, not frozen. Tandaan niyo yan, ha? Refrigerated viral specimens or yung mga specimens for viral diagnosis should be refrigerated, not frozen. Okay? Second, you need to process it within 12 to 24 hours. Okay? Pag hindi yan nagawa, most likely di-deplete yung value ng virus or pwede silang masira. Okay? Or mawala. So, ibig sabihin, magkakaroon ka ng false negative result. Pangatlo, you need to utilize the Rayon or the Dacron swabs. So, similarly sa mga bacteria and fungi, you need to utilize Rayon or Dacron swabs. The main reason here is yung swabs na to, number 1 sa lahat, they are sterile and meron silang mga additional nutrients, no, para mas lalong magtagal or ma-preserve yung buhay ng mga microorganisms. Okay? And lastly, we need to use VTMs. So, VTM means viral transport media. Sulat natin, viral transport media. Okay. So, naging common na, ito yung mga Rayon or Dacron swabs. Okay? So, ito naman yung VTMs. So, VTMs, they contain a liquid which is para sa pag-preserve ng mga virus. Okay? Meron din yung antibiotics. No? May mga antibiotics sila para mapatay yung mga contaminants that may intrude sa testing natin. And next, laboratory diagnosis. Okay, first one is cytological or histological studies. So, this one, nasa pangalan niya na mismo. So, as the name implies, cytologic and histological. Meaning, these are performed in the histopathology laboratory. Okay, or the histopathology section of the lab. So, gagamitin mo yung histotechnics mo, no? Para makapag-provide ng fixed and stained cells na babasahin or babasahin at i-interpret ng mga pathologists. Okay? So, usually, ang hinahanap ng mga pathologists dito, yung mga multinucleate giant cells or kaya yung mga inclusion bodies na makikita sa cytoplasm or sa nucleus. Once again, doon sa ating module 1, ano bang makikita? Sinong mga virus kasi yung makikita mo sa cytoplasm? So, these are the RNA viruses. Whereas sa nucleus, sino yung makikita mo? Yung mga DNA viruses. So, basically, yung mga pathologists, magkakaroon na sila ng idea if ever makita nila yung inclusions ay present dun sa cytoplasm nung tissue or nung slide, okay? O kaya dun sa nucleus. Once again, cytoplasm, RNA viruses, nucleus, DNA viruses. Next is the electron microscopy. Electron microscopy is not available in the routine clinical laboratory. Unang-una sa lahat dahil napakamahal nito, and this is the last resort kapag hindi mo talaga ma-identify yung virus na yun. So basically, you would only see this electron microscopy sa mga virology laboratories, sa mga research laboratory for viruses. So, dyan mo sila makikita ang electron microscopy. Okay? Ang principle ng electron microscopy is the negative staining. So, basically, light structures ng mga viruses against dark background. So, this is only for viruses who do not grow to cell cultures. Okay? Ulitin ko, electron microscopy lang ang pinaperform if that particular virus do not grow to cell cultures and unavailable tests for viral antigens. So, best example dito is noong unang labas ng novel COVID-19. Okay? Noong 2019, 2018. Back then, wala pang available antigen kits. Wala pang available testing. No? So, ang ginagawa pa lang nila nun is to collect the specimen directly from patients who are suffering from coronavirus, and then they put it in the electron microscope and then look at it, no? So, ayan. So, ito yung itsura ng coronavirus. So, makapapansin nyo, meron talaga siyang spike proteins, no? Letter C, we have the virus isolation in cell or tissue culture. This is considered as the gold standard for viral isolation. Ulitin ko, virus isolation in cell or tissue culture is considered as your gold standard for viral isolation. Okay? So, we have three types of cell culture. Number one, we have the primary cells. Pag sinabing primary cells, ito yung mga cells na isolated directly from human or animal tissue. Pangalawa naman, we have the transformed cells. Transformed cells are genetically modified para merong heightened sensitivity to stress or apoptotic signals to mga to. So, ulitin natin, primary cells isolated directly sa human or animals. Itong transformed cells, genetically modified sila. So, basically, galing din naman sila sa human or animal cells or pwedeng stem cells din sila. Pero, they modified it genetically para mas mapabilis yung reaction ng virus dun sa mga cells na yun. Mas mabilis yung tinatawag nating cytopathogenic effect sa kanila. Okay? And para mas ma-observe siya ng madali. Okay? Ulitin ko. Transformed cells, genetically modified para merong heightened sensitivity to stress and apoptotic signals. Ulit natin. Apoptotic signals. Okay? And third, we have the self-renewing cells. So, basically, immortal cell lines sila, no? So, ito yung mga stem cells na tinatawag natin. So, undifferentiated sila. Meaning, wala silang specific karyotype. Wala pa silang specific na tissues. Kasi tatandaan natin na ang mga viruses, meron lang silang specific na tina-target na tissues or cells na may receptors. So, basically, itong mga self-renewing cells, okay, stem cells lang sila. Kumbaga, sa bacteria, sila yung natawag na nutrient agar. Tapos, pwede mong dagdagan ng kung ano-anong mga enhancing reagents or mga enhancing media para mas maging siyang specific towards a particular bacteria. Okay? Sana magets nyo yun, no? Ulitin ko, pag self-renewing cells, stem cells sila, so immortal sila, na undifferentiated. No? Undifferentiated. Okay? Undifferentiated. So, ulitin natin, three types of cell culture. Primary cells, ito yung mas common na isolated directly sa human o animal cells. Madalas dito yung mga kidneys, no? Kidney cells. transform cells, mga genetically modified para mayroong heightened sensitivity towards stress, o yung tinatawag nating apoptotic signals. Okay? And lastly, we have the self-renewing cells or yung mga stem cells na undifferentiated na pwede mong palitan, okay, para maging kung ano man yung target tissue ng virus na kinukulture mo or ina-isolate. Okay? And usually, this cell cultures are animal or human cells grown in vitro in the laboratory. Okay? And then, syempre, ulitin natin, ginagawa ang cell culture to examine what we call the cytopathogenic effect. Tatakpan siya, so sulat natin cytopathogenic effect or yung CPE. So, cytopathogenic effect are those that are the cellular damage or changes in the cellular structure. So, kaya nga, cell culture, di ba? Kasi ito yung gustong tirahan ng mga viruses, and kapag pumunta sila dun sa particular cell na yun, sisirain nila or may gagawin silang changes dun sa cell na yun. So, ano yung changes na yun? Ano yung hinahanap natin or tinitignan natin na changes na yun? Ang tawag doon, CPE or yung cytopathogenic effect. So, ayan, example ng ating primary cell tsaka ng transform cell. So, primary cells, ayan, in-isolate sila from a particular tissue, and then nagkaroon ng contact towards the or against the viruses. Tapos, pwede kang gumawa ng tinatawag nating cell line. Okay? Sinasubculture natin yung yung primary cell na nag-contact with the viruses, tapos subculture to a what we call cell line. As transform cells naman, since they are already genetically enhanced para tumaas yung stress ng cells na to against the viruses, diretso mo na siyang pwedeng hanapin for the cytopathogenic effects. So here are some examples of cytopathogenic effect na makikita natin na mangyayari sa mga cell cultures, no, upon infecting with viruses. So pwedeng those cells become mag-rounding sila, okay, mag-clump, vacuolation, no, kaya magkaroon ng vacuoles dun sa loob ng mismong cell, granulation, giant multinuclear cells, cell fusion, cell destruction, or pwede rin lysis, sirain na mismo. Okay? Ngayon, take note of this. Sabi dito, no, yung cytopathogenic effect ng orthomyxo and paramyxo viruses, no? So, sila lang merong ganito, no? So, kailangan ka pang gumawa ng additional na procedure or process. Yan yung tinatawag nating hemadsorption or hemagglutination. So, basically, after mong masubculture sa cell line, okay, pwede mo siyang ipasa sa serology and pagpa-perform sila ng hemadsorption or hemagglutination. So, ito yung mga example natin. So, paramyxo, ayan, bukod dun sa hemagglutination, pwede rin siyang nagkaroon ng cinctia, tsaka basophilic cytoplasmic inclusion bodies. So, pox, inclusion bodies, herpes, nagkakaroon siya ng cytoplasmic stranding, tsaka nucleus, inclusion bodies. So, dito pa lang makikita nyo kung sino yung RNA, tsaka DNA dun sa kanilang inclusion bodies. Okay? Adenovirus, enlargement, tsaka grape-like clusters. Next, discuss naman natin about primary cell cultures. So, ito yung mga examples natin. We have the JK, RK, PMK, RMK, CMK, tsaka GMK. So, ayan yung mga transcription ng mga yan. So, primary cell cultures are to be subcultured pa to a tissue culture with at least same karyotype. Tandaan nyo yun, ha? Same karyotype. Okay? Ito yung tinatawag nating cell line. Kaya natin sila sinasubculture, no? So, cell line, ulitin natin, pag tinawag na cell line yan, kung anong specific cell na tatargetin ng virus, no? Yun na yung i-study natin for the CPE under the electron microscope. Okay? So, meron kang primary cell culture, and then kailangan mo yung i-subculture to a tissue culture with at least the same karyotype, and then ito na yung tinatawag nating cell line, which is the specific cell na tatargetin ng virus. Kaya lahat yan kidneys. Okay? Mapapansin nyo. Kidney sila. Kasi nga, ito yung pwede pang mag-differentiate sa iba. Kaya lang matagal. Okay? So, kailangan mo silang i-subculture to a cell line. And then, meron tayong tinatawag na ATCC. So, kung naalala nyo sa microbiology or sa bacteriology 1 or micro 1 or bacteriology, meron tayong tinatawag na ATCC sa mga culture media. Meron din tayo sa mga cell cultures. So, pag sinabing ATCC, these are the American type culture selection. Ito yung standardized na pang-isolate mo ng microorganism. Okay? So, say for example, hindi nyo na kailangan memorize, no? Example lang. Ang HEK, meron siyang ATCC na 293, RK13, and etc. So, okay? So, again, ATCC or yung American type culture selection, ito yung standardized na cell, kung virus ang pinag-usapan, no? Na cell culture for viruses or microorganisms. Next, direct detection of viral antigens or genes. Okay? So, meron tayong fluorescein-labeled monoclonal or polyclonal antibody, and yung mga yan, madedetect niyan through DFA or yung tinatawag na direct fluorescent antibody. So, ito yung resulta niyan, okay? Pwede rin through antigen-antibody complexes or through ELISA, gagamit ka ng machine, or pwede rin through latex agglutination lang. Okay? And ngayon, common na gumagamit din ng antigen kits. No? And all of these are performed under the immunology and serology section of the laboratory. Kasi nga, ang pinag-uusapan natin dito is antigen-antibody complexes or reactions. Okay? Okay, so here are some of the examples ng ELISA reagents, no, para ma-detect yung certain viruses. So, ayan, mga hepatitis. Hanggang dito, yan, hepatitis. Tapos ito, HIV. Tapos ito, cytomegalovirus. Tapos ito, COVID-19. Okay? Okay. Now, dito na tayo sa molecular diagnostic methods or tinatawag din nating mga nucleic acid tests. So, these are performed nowadays na ito na yung mas prefer ng mga physicians kasi nga sa immunology tsaka sa serological studies, indirect yung detection natin ng viruses kasi nga antigen-antibody reaction ang hinanap doon. Usually, antibody detection nga, IgM, IgG. Okay? Dito, technically, you are detecting the presence ng viruses exactly doon sa specimen ng pasyente mo. So, it has faster TAT or yung turnaround time. Ibig sabihin, ang mga results nito ng molecular diagnostic methods and nucleic acid tests is after an hour or after 2 hours, meron ka ng result. Unlike yung cell culture, which is really the gold standard. Kasi nga, ano talaga direct detection siya, and high specificity and high sensitivity siya. However, matagal siya because you need to culture the virus first. You need to subculture then para makakuha ka ng cell line. So, it takes around 2 weeks to a month para lang makakuha ka ng result mo. Whereas dito, sa molecular diagnostic methods, it has faster TAT. Sabi ko nga, 1 hour to 2 hours, meron ka ng result. Better sensitivity than the immunologic tsaka serological, no? And pwede ka rin makadetect ng multiple viruses simultaneously. So, meron siyang listahan, tas makikita niya na or madedetect niya kung sino doon yung present na virus, kung meron man. Okay? So, for the examples, we have the RFLP or yung restriction fragment length. Dito, DNA comparison ang ginagawa. Okay? Pangalawa, we have the DNA and RNA genetic probes. So, it detects and locate the specific genetic sequence. Okay? Hahanapin niya yung specific na DNA or RNA virus genetic sequence. Mamaya may papakita akong picture na familiar na kayo kasi diniscuss din natin to dun sa fungi diagnosis. Okay? So, pangatlo, we have the PCR or the polymerase chain reaction. So, for DNA viruses to, tandaan nyo pag sinabing PCR, polymerase chain reaction, DNA viruses. Pag naman reverse transcriptase PCR or reverse transcriptase polymerase chain reaction, it detects RNA viruses. And lastly, we have the real-time PCR. So, real-time PCR ang tinatawag na nucleic acid test for HIV genome viral load. So, dito, quantitatively, titignan niya yung HIV genome viral load. Okay? So, titignan niya kung gaano karami or gaano yung amount, yung specific amount ng viruses ng HIV na present sa pasyente natin. So, usually, ginagawa to sa mga malalaking laboratory na capable mag-screen ng mga blood bags o yung mga donor bags, no? Yung mga dugo na transfuse, no? So, ang ginagawa nila is kailangan nilang ma-detect kung gaano karami yung amount ng HIV doon sa specimen na yun. Okay? So, this is an example of the real-time PCR. Okay? Nucleic acid testing for HIV. Okay, so this is an actual example of the film array torch. Ayan, wherein nag-run ako ng actual specimen. So, ayan. So, for example, this one is the GI panel or the gastrointestinal panel. Hahanapin ng machine na to yung exact DNA or nucleic acid na present doon sa specimen. So, for example, this one, na-detect niya na merong Clostridium, which is a bacteria. So, basically, kaya niyang mag-detect ng DNA or RNA na present sa bacteria, present sa, bacteria din to, E. coli, na may specific strain, tas parasites, and as well as viruses. So, dito naman sa meningitis panel, ME panel, So, again, pwede nyo makita yung DNA or RNA genome sequence ng bacteria, viruses, and ng yeast, which is yung Cryptococcus neoformans. So, dito, na-detect niya na may presence ng nucleic acid ng herpes simplex virus 1. Another machine na gumagamit ng principle of the PCR and RT-PCR is the GeneXpert, which was also discussed noong fungal lab diagnosis. So this one, na-detect niya naman yung presence ng HBV, no? Hepatitis B virus. So ito naman is specific lang. Iisa lang yung kaya niyang i-detect pero with high specificity, and lalagyan niya pa ng specific value kung ilan yung viral load na present. So these are other pictures. Meron tayong film array na mas malaking version kasi mas marami siyang kayang i-run na panel ng sabay-sabay. Okay. This one is the RT-PCR. Tapos ito, PCR machine. And last is the serologic detection of viral antibodies. So this one, same goes with the immunologic, now indirect indicator that infection or exposure to the virus has occurred in the individual's past or current o kaya past infections ang hinahanap dito. Okay? So, pag hinahanap ng physician is virus-specific antibody lang. Ibig sabihin kung may presence ng antibody during the infection, ang magko-collect ka 1 to 2 weeks after the symptoms kapag IgM yung hahanapin niya. Pag IgG naman, 1 to 2 days after the primary viral infection. Again, pag sinabing virus-specific antibody, ibig sabihin, ang hinahanap niya is yung presence ng antibody. Okay? Gusto lang malaman ng physician mo if positive ba siya sa ganitong virus. Okay? Sa mga, kunwari, meron tayong, ito yung most common yung dengue. Yan. Meron tayong dengue NS1, IgG, and IgM. So ito, kit lang to, and then sasabihin niya kung positive or negative. Wala siyang quantitative value na ibibigay. So basically, kapag nag-positive yan sa NS1, meaning, bagong-bago pa lang yung kanyang infection with the dengue virus because nasa dugo niya pa or nasa system niya pa yung antigen of that particular virus, ng dengue virus. Whereas, pag nag-positive siya sa IgG, ibig sabihin, meron ng days, no, na na-infect siya ng dengue. And pag nag-positive din yan sa IgM, ibig sabihin, weeks na yung infection niya ng dengue. Okay? So, example, pag yung NS1 niya, positive, IgG, IgM, negative, ibig sabihin, bagong-bago pa lang. Kapag naman NS1 positive, IgG positive, IgM negative, ibig sabihin, days pa lang. Pag naman, sir, pwede ba yung NS1 negative tas nag-positive sa IgG at IgM? Yes, pwede din yun. Pwede pa rin yun because may possibility na mabilis mag-react yung kanyang antibodies and natanggal niya agad yung dengue virus sa system niya. However, dahil na-infect siya ng dengue, there's still yung symptoms pa rin is lilitaw. Okay? So, next. Pag naman ang hanap ng physician mo is peak levels ng antibody, ibig sabihin yung highest quantitative value ng mga antibodies na to, kung yun yung kailangan hanapin ng doktor, meaning, ang collection mo ng kanyang serum would be For IgM, 3 to 6 weeks. Okay, peak levels yan. Pag naman sa IgG antibody, 4 to 12 weeks. Okay? And for serologic detection, meron tayong tinatawag na paired sera. Paired sera, sa pangalan niya mismo, meron kang dalawang specimen. The first specimen is the acute phase specimen. Ito yung i-collect mo yung kanyang serum after the clinical signs first appear. Okay? Pag unang lumitaw yung symptoms niya ng viral infection. Okay? Yun ay acute phase specimen. Yung second specimen naman is i-collect mo yan after 2 to 3 weeks later. Pero it depends sa virus pa rin. Okay? Ang tawag natin dito is the convalescent phase specimen. So, itong paired sera na to, ano purpose niyan? So, ang first specimen mo, nagkaroon siya ng value. So, for example, purposes lang, no? 2, kunwari 2 yung kanyang value. Ngayon, kapag ang convalescent phase specimen niya naging 8, okay, ibig sabihin, seropositive ang inyong patient. Anong ibig sabihin pag seropositive reaction? Seropositive reaction means merong 4-fold increase in antibody titer. Sulat natin, 4-fold increase in antibody titer. Ang tawag dito, seropositive reaction. And kapag may seropositive reaction, it means meron nga siyang viral infection. Okay? Tandaan niyo yun, no? Paired sera, acute phase, convalescent phase, and kapag nagkaroon ng 4-fold increase in antibody titer, meaning seropositive reaction. And seropositive reaction means merong viral infection. And that's it. Kapag may questions kayo, just email me. And that's it. Thank you for listening and God bless.